Hello, good evening. Can anyone confirm that my voice is clear and everything is perfect? Can anyone confirm, please? Yes, sir. Good noon. Manav Bhavesh, welcome back. Thank you, Bhavesh. Thank you, Manav. Is it clear? My voice is clear. Is my voice is clear? बताओ कोई बताओ. Okay. Then जल्दी फटाफट शेयर कर दो. ठीक है, सबको रीच होना चाहिए. At least for your friends who are preparing for this ICAR PhD exam, okay, in the stream of genetics and plant breeding or Tech, maybe. One thing, Manav Pate sir, also important. No. In this particular video, I am going to discuss part C of genetics and plant breeding. ठीक है, पार्ट बी भी मैं देखा था. उसमें तो उतना डीप क्वेश्चन और उतना सोचने के लिए थोड़ा उतना बड़ा डिफिकल्ट क्वेश्चन तो नहीं था, इसलिए मैं बस पार्ट सी डिस्कस कर रहा है. इसके बाद ठीक है, इसके बाद देख के पार्ट ए पार्ट बी भी नेक्स्ट वीडियो में करने के लिए कोशिश करता हूँ. ठीक है, वही आई एम टेकिंग ओनली पार्ट सी. इट कंटेन्स 50 क्वेश्चन. Definitely it needs minimum 1.5 or 2 hours to discuss this particular 50 questions, since these questions are very difficult. Okay, and also everything will be new sometime, so it will take too much time. This is not about one day preparation. I have sit for 3 days to analyze each and every question, so that it can be discussed with proper explanation. Okay, this is as I told, once again I am telling, this is for part C. Okay, part C, ICAR PhD genetics and plant breeding in 2025. This is regarding 2025 paper. Okay, only 8 have joined now. There are so many people in our group, they don't want it, I think. They will ask PYQ series for PhD sir, marathon for PhD sir, then no one wants to join. ಗುರು ಬ್ರಾಡ್ಕಾಸ್ಟ್ ಮಾಡಲ್ಲ ಮಾಡಿದ್ಯಾ? Okay, 14. At least minimum 30, 40, 30, so that we can start our session. It is very informative. Definitely you guys will like it. Okay, we can see how it goes. Okay, then we are not going to waste any time now. We will start with our first question. Anyway, you guys already know. Just I will give once again. I am Abhishek. I completed my undergraduate from US Bangalore. I completed my masters from ICAR IR New Delhi with All India rank 21st. Then I got All India rank 4 for ICAR PhD. I am currently pursuing my PhD in the same division of genetics, ICAR IR New Delhi. I also cleared ASRB net and joined CSI UGC net. Recently, I have visited Greece and Italy for one of my PhD summer school. This is all about me. Okay. Sir, one grand mark test if possible before exam, sir. Okay, we will try to give. Okay, we are thinking of it. Sir, seed science paper question are also included. Teja, in this paper, as I told, I am covering part C. Okay, anyway, this is my first video for PhD exam. So I will think about it in the next class. Okay, the part B section. Okay, for now it is all about part C. Okay, in the genetics and plant breeding. So this is the first question. Okay, they have asked in part C. Statement 1, progressive heterosis is an objection to the dominance hypothesis for heterosis. Statement 2, progressive heterosis can be observed only in autopolyploid species. Yes. Can anyone? What is the answer? What will be the answer according to you? First statement is progressive heterosis is an objection to the dominance hypothesis. Okay. The second option is progressive heterosis can be observed only in autopolyploid species. Bhavesh, C. Statement 1 is true, statement 2 is false. Yes. Your progressive heterosis is one of the objection for the dominance hypothesis. Dominance hypothesis contains, there are 4-5 objections. We will discuss what exactly those are. And in statement 2, they are telling that it can be observed only in autopolyploid species. It's not like that. Okay. You can expect that progressive heterosis in any polyploid species, not only autopolyploid species. What exactly the progressive heterosis? Okay, everything we can see. Someone is telling both are true. No. Anshul. Okay, it's not that option. As I told, we can observe that progressive heterosis in any polyploid species, not only auto. What are the objections here? First one, in dominance hypothesis, okay, it is given by Davenport. Failure to isolate inbreds as vigorous as F1. What is dominance hypothesis? It is due to the masking effect of recessive allele by the dominant allele. Okay, so that your both capital A and capital A small a, these both are having equal effect compared to small a. Okay, this is what dominance hypothesis will tell. So what is the first objection? Failure to isolate inbreds as vigorous as F1. If your dominance hypothesis is true, okay, then you can isolate an inbreed which is having like this. If your inbreed having the combination like all are in homozygous condition, then it is vigorous as your F1. Capital A, small a, capital B, C, D, E, B, C, D, E. Okay, it is vigorous as your F1, but it will not happen like this. Okay, it is one of the objection, but it is explained by Jones. Okay, it is explained by the Jones that dominance of linked gene hypothesis. Okay, dominance of linked gene hypothesis. What he told that each homozygous genes for dominant homozygous genes may be linked with recessive homozygous genes. Okay, like this: capital A, capital B, small c, small d, small e, small e, then again capital F, like this. They have linked with the repulsion phase, so that it is very difficult to expect or to isolate complete homozygous plant. Okay, it is explained by Jones as in 1917 as dominance of linked gene hypothesis. Okay, the second objection, symmetrical distribution in F2. Okay, what is symmetrical distribution? You can see this is symmetrical normal distribution. Okay, but according to dominance hypothesis, you can see this will be the ratio 1 to 1 in case of F2. Okay, since these two are giving the same phenotype, then it should be 3:1 as per your dominance. If it is 3:1, then you should expect what? 3:1, like this, the skewed distribution in F2. But you will not expect this skewed distribution. You will always get symmetrical distribution in case of F2, like this. This is normal or symmetrical distribution. But you should have expect this 3:1. Okay, it is also explained by Collins in 1921 Okay, due to the more number of genes controlling the trait, it is not like single 3:1, see, not a single gene. Okay, since it is controlling by more number of genes, okay, your distribution will be affected by environment. So this skewed distribution will convert into symmetrical normal distribution. Okay, sir, when will you call all? सर व्हेन विल यू कॉल PYQs पर SST? अरे भाई मैं तो SST में SST से नहीं है भाई हाँ, सीड साइंस और टेक्नोलॉजी से नहीं है, ठीक है। हम जेनेटिक्स एंड प्लांट ब्रीडिंग का देख रहा है अभी, पार्ट सी का, ठीक है। पार्ट बी में सीड टेक आता है। ऑलरेडी आई हैव टोल्ड दैट if anything related to CTech or any other subjects, we will think about it. First, anyway, we started it. First we will cover genetics and plant breeding. Okay. Then the magnitude of heterosis. After evolving or after improvement of each inbred, okay, they will come into homozygous condition. Maybe they are in the dominant homozygous or recessive homozygous, we don't know. When you are going too much inbreeding, okay, so the magnitude of heterosis should be decreased according to dominance hypothesis. Okay, since it will convert into homozygous recessive also. But okay, as we saw in our cases, after some decades, after 10 years, 20 years, the heterosis is improving. The magnitude of heterosis is improving. It is also one of the objection for your dominance hypothesis. Next, progressive heterosis in case of tetraploids. Okay, what is progressive heterosis? It is nothing but, for example, if I take any tetraploid, A1, A1 and A2, A2. Okay, the cross between A1, 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 A1 into A2, 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 A2 will be your A1, A1 and A2, A2. If we compare this particular F1 with A1, A2, A3, A3, this particular performance will be superior than this one, since it contains 3 divergent alleles. Here only 2. According to dominance hypothesis, should be this one should have superior performance, but see, due to the divergent alleles in the different combination, okay, it will give superior performance. Okay, this we will call it as a progressive heterosis in case of tetraploid. Okay, if you guys have any doubts in between, okay, you can just put it in comment box. Please tell Hari sir to get it done. ठीक है, हरी सर तो पता नहीं क्या क्या करना है, जेनेटिक्स का कर रहा है, मैराथन सीटेक का भी करना है, ठीक है, मैराथन करना है, उसके बाद ठीक है PYQ भी देखेंगे. नेक्स्ट, फास्टर इनब्रीडिंग डिप्रेशन इन टेट्राप्लॉइड्स. ओके, in tetraploids what will happen, there will be a faster inbreeding depression compared to homozygosity. Okay, if it is controlling by any only in case of diploids, okay, the two alleles. Okay, what about the inbreeding depression and the homozygosity? It will be same. There will be each and every generation there will be a decrease in 50% of your heterozygosity and increasing 50% of homozygosity. But in case of tetraploids, it will not happen like that. Okay, your inbreeding depression will be faster compared to the recessive homozygous. For example, if you are, this is your allelic combination. For example, A, you can take in a tetraploid, A, and another is small a, small a, small a, small a. Okay, when you will cross, you will get capital A, small a, small a. Okay, this is the condition in case of tetraploid, F1 condition. If it is go to F2, then there will be different combination, since it is having 4 copies. Okay, out of 18, I think 18 genotype, you will get only one small a, small a, small a, small a, this condition. Okay, like all will be in recessive condition. It will be very less frequency. If you compare this one, when there are having 2 genes, your F1 will be what? Capital A, small a. Your F2 you will get in out of 4 Okay, one will be small a. You can see the homozygosity will be higher frequency in case of your F2, and inbreeding depression also it will be similar. But in this case, okay, when they are when in autotetraploids, okay, the condition of this will be the frequency will be very less compared to inbreeding depression. Inbreeding depression will be very faster. Okay, if we have very two similar genes, there will be inbreeding depression will happen. But the homozygous recessive, the frequency will very less. That's what your fifth objection. Okay, faster inbreeding depression in tetraploids. Okay, it will not match with your recessive homozygous condition. Okay, this is about the first question. Okay, this, the different objections. Okay, then in the CRISPR-Cas9 system, the genome editing the role of scaffold region of a single guide RNA. Okay, what will be the role? Yes, just ABCD option in the chart box. Yes, Bhavesh. Anshul. Yes, come on. What is the role of single guide RNA? Associated with and activates the Cas9. Okay, we will see the components of your CRISPR-Cas9. Okay, this will be your complete single guide RNA. Okay, the first in the natural condition it will be CRISPR RNA. Okay, then we have made it the single guide RNA. Okay, with CRISPR RNA and tracer RNA we have merged with. Okay, now we are artificially we are made it only single guide RNA. What it will do? It will helps to associate and activates the Cas9 enzyme. Okay, it will having guide RNA so that it will go and bind target sequence. Okay, and it will also helps in the attachment of Cas9 enzyme so that there will be a precise cut in the genome editing. So your answer will be associate with and activates Cas9. The next question. The ratio of the number of null hypotheses wrongly rejected to the total number of null hypotheses rejected in the experiment. Yes? Yes, come on. The ratio of number of null hypotheses wrongly rejected to the, yes, the false discovery rate. What exactly the false discovery rate? The number of false positive. That what that means? Okay, there is a null hypothesis. What null hypothesis will explains? There is no significant difference. This is your null hypothesis. But what you are doing? You are rejecting that null hypothesis. If you are rejecting null hypothesis, that means you are telling that there is a significant difference in the experiment. So that means you are taking as a positive, as a false positive. Okay, there is no coronavirus in case of your test material, but you are telling that there is a coronavirus. That means it is false positive. Okay, you are rejecting the null hypothesis. What is FDR? That rate, the rate at which you are doing this particular mistake. The number of false positive to the total number of rejected null hypothesis. Sometimes you will genuinely reject the null hypothesis. Okay, out of those total rejected null hypothesis, how many are false positive? Okay, false positive is nothing but it occurs when the null hypothesis actually true, but you are incorrectly rejecting it. Okay, that you are telling that there is some significant difference, so that you are making a false positive. Okay, this we will call it as a false discovery rate, the ratio of wrongly rejected null hypothesis to the total rejected hypothesis. The eukaryotic 5.8 srRNA is constituted of how many nucleotides? Yes, the eukaryotic 5.8 srRNA is constituted of, बताओ D or A? It is again they have asked from the basic genetic part. Okay, you can expect these kinds of questions sometime. Okay, there will be some of out of your box. Okay, Shinchan इधर भी आ गया क्या? अरे भाई Akshita, yeah. Yes, this is about 160 nucleotides. In case of size of eukaryotic rRNAs, 18S. Okay, 18S will be your 1800 nucleotides. 5.8S is having 160 nucleotides. Okay, there will be a different number of nucleotides. In case of your prokaryotic RNAs, your 16S RNA will be 1540. Your 5S RNA is 120. Okay, here 120. The same for 5S RNA, but 5.8 S RNA will be 160 nucleotides. Only 6. Again, I am telling this is very important. Okay, I will not get this much information anywhere. Okay, जल्दी शेयर करो फटाफट. Yes. Type Bhavesh type 1 error. Right? Type 1 error is nothing but you are rejecting null hypothesis when it is true. Okay. There is a significant difference. There is no significant difference. This is your null hypothesis. Okay. Yes, it is true, but you are rejecting it. That means false positive. That's what your null hypothesis. Sorry, the type 1 error. Bhavesh. Matlab you are asking that type 1 error was type 1 error. Sir, will you solve all papers from GPB SRF? अरे भाई मैं वही बताना था अभी. बस 10 लोग 12 there are only 10-12 members. Okay, and they are going, they are coming. It, as I told, it will be always based on your interest. Okay. Kedar Patil, ಊಟ ಆಯ್ತಾ ಅಣ್ಣಾಜಿ? ಊಟ ಆಗಿಲ್ಲಪ್ಪ, ಇದೆಲ್ಲ ಮುಗಿಸಿ ಊಟ ಮಾಡೋಣ, ಸರಿನಾ? So this is about the next. Then the next question is, the paternal applied induction in maize is controlled by not like dad gene. The statement 2, the not like dad gene codes for an specific phospholipase. Yes, Bhavesh. Yes, बताओ. Kedar, बताओ. Both statement 1 and 2 are incorrect. Exactly. Your paternal applied induction in maize is controlled by not like dad gene. Okay, your not like dad gene, that is your NLD, is not paternal applied induction. It is maternal applied induction. Okay, it will induce the maternal applieds. When you cross with these particular lines which are having NLD gene, okay, it will induce the maternal applied induction. That means we can use this NLD line as a male parent. Okay, since it is inducing the female haploids. The not like dad gene codes for an specific phospholipase. If it is maternal applied induction, okay, then it is producing maternal haploids, so that there should be a no contribution from the male side. So it should be pollen specific. Okay, this is not specific. This is pollen specific phospholipase, so that it will kill the pollen grains, so that there will be only induction of maternal haploids. Okay. IG1 control paternal, sir. Good. Kedar is a agronomy PG aspirant. How will he answer GPB PhD question? If you are agronomy PYQ aspirant, then why you are wasting your time here? जाओ भाई पढ़ाई करो. कंपटीशन है ज्यादा agronomy में plant science के कंपेयर करना, तो ठीक है. Yes, Bhavesh, pollen specific phospholipase, so that your both the statements are incorrect. If you want to see the maternal applied induction and paternal induction, the difference, Here in maternal applied induction, your applied genes come from female parent, as I told. Okay, the maternal applied induction, that means you are inducing the female parent applieds. In paternal applied induction, you will induce male parent. But the lines, okay, you have what you are going to use. Okay, you will, in case of your maternal applied induction, the lines you will use as a male. In paternal applied induction, the lines which are having the induction capacity, you will use as a female. Okay, the main gene sylvanoids are maternal, lineal gene. You are not like dad gene. Okay, these are the genes, and IG1. Okay, this is indeterminate gametophyte. This is from paternal applied induction. The stock 6 is very important maternal applied induction line. Okay, the famous, they are asked frequently. As I told, the defects where it will try to induce pollen function, paternal genome elimination. Okay, the genes which are the lines. Okay, they have associated with paternal pollen function or paternal genome elimination. Here, in case of your paternal applied induction, the female gametophyte development will not happen. That's why it is indeterminate gametophyte. There is no female gametophyte formation. So everything will be from male parent. Okay, maternal applied result, here paternal applied. These are the examples, other examples for your maternal applied induction. Okay, the example for your paternal is IG1. Don't confuse. Okay, IG1, the only one, the gene and the line is responsible for paternal applied induction. All other we can see in the maternal applied induction. Okay, you are inducing the maternal side to the applied. Okay, clear? Need soil science PhD PYQ session. Okay, I will convey these things to the respective mentors. Okay, clear? Okay, only Bhavesh is responding. What about others? What about our students? Kaha chala gaya sare? The x-axis of the Manhattan plot shows SNP being sorted by chromosome location. The statement 2, the y-axis of the Manhattan plot shows positive logarithm of p-values. The statement 3, y-axis of the Manhattan plot shows negative logarithm of odd values. Which are the statements are correct and which are incorrect? Yes? Yes, anyone? Your x-axis of the Manhattan plot shows SNP in the being sorted by the chromosome. It is right answer. Okay. Then the statement 2, the y-axis of the Manhattan plot show positive logarithm of p-values. It will show negative logarithm of, okay, the negative logarithm of odd values. The statement 3, y-axis of the Manhattan plot shows negative logarithm of odd values. That is, they are telling that negative log values. Okay, there should be a negative logarithm of p-values. So your statement, before that we will see what exactly the Manhattan plot is. It will comes in your GWAS or association mapping. The SNPs which are associated with the target trait. Okay, we will decide based on this Manhattan plot. Why we will call it as Manhattan plot? Okay, I have already explained in our live classes. Since it will look like a Manhattan city. Okay, your y-axis contains negative logarithm of p-value. P is probability value. When you will apply the different probability value, okay, if your probability value will be very less, then your negative logarithm will be very high. Okay, that is the basis of your log values. Okay, it will convert the most small digit values into the constant number. For example, if it is 0.0005, it is your, if you take the probability value like this, then you will get some negative logarithm of that p value. Okay, if it is going less, your log p value will be more. That means the probability of the error, it is too much less, then your negative logarithm value will be more, so that your SNP which is associated with the trait, the chance will be very high. You can see, if you keep on increasing negative logarithm of p value, you will get any one of the SNP, that you can tell that it is strongly associated with the particular trait which you are going to analyze. Okay, it is located in the particular chromosome, like chromosome 6. This is the chromosome location and this is the negative logarithm of p value. Okay, then your answer should be only the statement 1 is true. Statement 1 is true and the statement 2 and 3 are false. Okay, bone for any correction is this only, isn't it, sir? Bone for any correction is how you can decide this particular 5. Here we have took a 5 as a cutoff value. Okay, it is then we will plot the graph with the negative logarithm of p values and the chromosome, the different SRP markers. After that you should go for the cutoff, so that you can only take this as your SNP. Okay, that is based on the cutoff what you will decide, that is your bone for any correction cutoff. Okay, it will be based on the total number of markers you have decided with the probability value. Okay, the probability value and with the respect to the number of markers. How many number of markers you are going to analyze, based on that the total number of markers and the p value at what you are going to discuss, so going to decide. So based on these two you will go for a bone for any correction. Okay, the next question, which of the following statement are correct with respect to the speciation? The geographical isolation is essential for allopatric speciation. Sympatric speciation occurs without geographical isolation. Polyploidy is an essential for allopatric speciation. Random genetic drift and selection are the main differentiation forces in allopatric speciation. Okay, what are the correct statement with respect to speciation? Yes, anyone? Yes, come on. First, if you want to solve this, first you should know what is allopatric and sympatric speciation. This is again, this is the basic speciation the definition explanation I have discussed in ICAR PG class itself. Okay, the simple thing, in case of sympatric speciation, okay, it will be happen in the same place. There is no geographical barriers. That means there are no like it is something is happening in the Asia and something happened in the America. Okay, everything will be in the same geographical location, but the barriers are like polyploidy or any chromosomal doubling. These will create the barriers between two genotypes. Okay, after some 10, 15 or some 100, 150 years, they will be going to separate. They will going to be a two different species. Okay, the barrier will be here some any genetics or any chromosomal barriers. Okay, in the same location. But in case of allopatric speciation, okay, I will go to any one of the potato growing region that is very popular in case of Europe or in case of South America. I will bring some genotypes from there. Then I will start to grow here. After some 100 years, here it will be due to the different climatic condition, different temperature and rainfall. This species will going to be the different species, one which is having original species in the Europe region, the geographical barrier. Okay, your geographical isolation is essential for allopatric speciation, and the sympatric speciation occurs without geographical isolation. Yes, it is also correct. The polyploidy is an essential mechanism for allopatric, as I told. Okay, for allopatric, it is due to geographical barrier, not due to polyploidy. Okay. Then random genetic drift or selection are the main differences over allopatric. Yes. What I have did, the selection. What is random genetic drift? It is due to founder effect, bottleneck effect. Okay, what is founder effect? Out of the original population, some of the species will move to the another place. and they will make a new population. That is your founder effect. This is also example for allopatric speciation. There is a geographical barrier. They will move to some other place and there will be a speciation. But in case of bottleneck effect, due to some adverse environmental condition or anything, there will be a loss of some individuals in the population. So the frequency will change, the allelic frequency. Okay, these are two different effects. With that, the founder effect will be more suitable for this random genetic drift. Okay, your founder effect will be the one of the force for allopatric speciation. So A, B and D. A, B, D only. Okay. Yes, good. Ananna Patra. Yes. The next question. Endonuclease and exonuclease are involved in the generation of major matured eukaryotic RNAs by cleavage and trimming the precursor rRNA. This statement will correctly framed. Okay, your exonuclease and endonuclease are the responsible to make it matured RNA. Okay, from the eukaryotic rRNA through the process of cleavage, trimming of that precursor RNA, it will make into mature rRNA. The precursor rRNA contains the sequence of the 18S, 5.8S, 28S rRNAs. Again, it is true. Okay, your 5S RNA will be separately processed. Okay, remaining the 18S, 5.8S and 28S rRNAs are the coming from your precursor rRNA. Yes, both the statements 1 and 2 are correct. Right, what about others? Okay, you should be very attentive and you should be like answer your expected answers. Okay, in the chat box. Okay, as I told, okay, the future classes will be based on the interaction and your interest and participation. Okay, the next question. The linkage disequilibrium information is helpful. Okay, in determination of market rate association, studies in population genetics, analysis of effects of domestication, and demographic history of plants. What is linkage disequilibrium? It is non-random association between the one or the two or more loci. Okay, or two or more alleles of a loci. It is a non-random association. Okay, what is random association? Which is exactly you can see in with the law of segregation and law of independent assortment. Okay, there is a non-random association between the alleles, so that there will be a difference in the frequency of the final alleles. Okay, so the LD will give market rate association. In case of GWAS, we will use LD. Studies in population genetics, yes, whether it is in the Ardivar equilibrium or not, we can use LD. Analysis of the effects of domestication, definitely. Okay, how they are domesticated, they are having some historical recombination or not, they are moving together, the alleles are moving together, maybe there is a presence of linkage or not. Everything we can get information from LD. Demographic history of plants. Okay, again it is based on LD. Okay, your A, B, C, D. Okay, the option 3. The next question. The nitrogen content of protein is approximately, it is direct question, which is they have asked in the part C. Okay, the 16%. The protein, sorry, the nitrogen content in the protein. Okay, this is the general question. I don't know why they have included in the part C. Next, the precision of QTL mapping is directly proportional to the size of the confidence or support interval. That defines the genomic position of a QTL and to the standard error of the genetic effects of QTL alleles. Your statement 2 is talking about accuracy of QTL mapping. It is closeness of the estimates of QTL location and the size of the QTL effect obtained from the study with the true values. Okay, study to their true values. What is precision of QTL mapping? How much precise you are to that particular QTL. Okay, It is not directly proportional, okay, of the size of the confidence or support interval. I have explained this in our live classes. The confidence interval, okay, where there is a probability of that particular QTL. If it is too much wide, your confidence interval, then how you can tell that it is precise QTL mapping? Your confidence interval should be minimum so that you can try to dig deeper to get a probable genes, right? It should be a with respect to the size of the confidence or support interval and the position of the QTL and the standard error for the genetic effects. The both the standard error and the size of the confidence interval should be minimum, okay, so that it is not directly proportional, it is indirectly proportional to the size of the confidence interval and the standard error. There should be a minimum size and minimum standard error. Then we can tell that it is precision of QTL mapping. Then accuracy, as I told, as this statement is telling, the closeness of the estimated QTL values or size of the QTL to their true values. That is accuracy. This is the statement 2 is correct, okay, the true. But your statement true is, so the statement 1 is false. Which one? The option D. Yes, Bhavesh, inversely proportional. Good. What about the next question? The breeding behavior of monosomics wheat indicates the percentage recovery of disomic plants upon self-pollination. Okay, the monosomic is 2n minus 1. What is the breeding behavior? If you know the breeding behavior of monosomics, okay, you can tell the answer. Yes. Let's see. Who will tell the yeah, you can tell the answer, maybe based on your key answer or whatever it may be. I need explanation. If you want to tell in a single line the reason which you are getting, yes, the answer will be 24%. Okay, I will agree. Okay, now I have a gift for you guys. If anyone tells that, okay, why you are getting 24? Array, what is the breeding behavior? 2n minus 1 of plant breeding behavior. You will get two types of gametes. One is n, one is n minus 1. Okay, n minus 1. In case of your female side, n minus 1. 2n minus 1. Here also 2n minus 1. Here 2n minus 2. Okay, this is normal disomic, monosomic and nullisomic. This will be the different condition. Okay, there should be a like 50-50, 50-50, 25%, 25%, 25%, 25%. Your disomic will be 25%. Okay, disomic 25%. This will be your what? Monosomic 50% and nullisomic will be 25%. But it is not happening like that. There will be a different combination. You will get the different percentage of frequency in case of wheat. Yes, anyone? What's the reason for that? Pollens can't tolerate aneuploidy. Egg can tolerate more amount of aneuploidy. Yes, what you are telling is exactly correct. What will call for that? There is one term. That's what I am expecting, Bhavesh. There is a term for this particular situation. Okay. In wheat, that particularly in wheat, okay, whatever the crop is, okay, as you told, pollen can't tolerate aneuploidy. There is a problem with the pollens, but egg can tolerate more amount of aneuploidy. Okay. What is the term for that? Meiotic drive. No, I am expecting something else. Which is related to, okay, anyway. Univalent shift. No, Shashank. No, Shashank. There is a term, okay, you can after uploading this particular video in your YouTube, okay, once the live session ends, At that time also you can answer. Okay, I will see the comments. Then if you have any, if you guys given any right answer, definitely I will send you a gift. Okay, I will see the comment box. Okay, there is a term for this particular condition. It will be not in case of your gametes will not comes in case of 25 sorry 50 50 50 50. As Bhavesh told, there will be some deviation. Why that deviation? Okay, there is one term. Okay, we will see who will give. But in particularly in case of wheat, okay, there is a deviation. How much deviation? Okay, particularly in wheat, your male side, your N gametes will 96%. Your N minus 1 will be 4%. In case of female side, N will be 25%. Your N minus will be 75%. 96, N minus 1, 4%. If you multiply 96 into 25, you will get 24%. 4 into 25, 1%. 75 into 96, you will get it around 72, and 75 into 4, 75 into 4, around 3%. Okay, this will be the condition. Your 24% will be disomics. They are asking disomic, right? Yes. You are 72 and 1%. 72 and 1%. Okay, these two will be monosomics. And about 3% will nullisomics. This will be the your final frequency. That too in case of wheat. In every crop there will be deviation in case of your monosomics, breeding behavior. But particularly in wheat, they have estimated it. Okay, it will be like this. Okay, clear? Kuch to gamete viability tha sir. There is a particular word I am telling. Okay, again I am telling. Okay, you can put it in comment box. Okay, after the completion of class also. Okay, I will definitely send you a gift because of no. There will be different kinds of word in case if you go through the molecular mechanism, but there will be some generic name. Okay, which again I am giving an hint. It is related to pollen. It is related to pollen. Okay. The next question. The neck color of a bird is controlled by pair of codominant allele in such a way that your homozygous condition will produce red color neck. Okay. Your homozygous recessive are in another allele white color. When they are in heterozygous, they will produce pink color. An independently segregating locus governs the length of the feathers. There is another allele, okay, which is controlling the length of feathers. FF possesses normal feather, heterozygous will globe feather, and homozygous will lethal. Okay, this is the condition. They are asking what will be the frequency of pink necked. If it is coming pink necked, what is the condition here? You can see red, white, and pink. Your pink will be NR, NW. Okay, then koi laga diya already answer. We will call that as a pollen serodation. Okay, your pollens in this condition, why you are seeing only 4% or N minus 1? Since it is not having one of the chromosome, okay, the abnormality, there is a competition between the pollens which are having N and which are having N minus 1. Okay, maybe they are producing in the equal contribution during gamete formation, but when they are having breeding with the female alleles, okay, only the 96% of the pollens which are having N will germinate. Due to the competition, the N minus 1 pollens will not germinate properly. Okay, if they germinate also, there will be a pollen burst, or due to competition, they will lose through the breeding in the breeding mechanism. Okay, pollen serodation. Shashank, कहाँ से आप? Where are you from? Shashank Biradar. You are from our course or you have enrolled in any of our courses? Shashank? Pankaj, as I told, there is a term. This is given by Shashank Biradar. अरे भाई पूछ रहा है, बताओ शशांक, where are you from? Did you purchase? Yes sir, course student. Okay, then if you are our course student, you know how to contact me. So just send your address so that you will get an gift from my side. Okay, Shashank. Then, okay, this is over. This is the condition. Okay, before that you can type your answer in the comment box. Okay, we will see you have given the right answer or not. Okay, side by we will see the answer also. Pink color. Then they are asking pink colored and normal feathered birds. Normal feathered birds means yes, FF will possess normal feather. This is the genotype. The frequency of this genotype they are asking. Okay, I solved it, sir. 2 by 12 or 1 by 6. B option. Okay, this is the condition. Now, if you want this particular allele combination, okay, if you take this particular allele combination, this will be the 1 by 2. The particular the genotype out of N R NR NR NW NW NW. Okay, out of these three combination, okay, if you want to get this particular NR NW, that is 1:2. This is 1 by 4, 1 by 4. Okay, this is 1 by 2. Then what about this capital F, capital F? Okay, yes. What you will think, this is 1 by 4. This is like homozygous condition. This is 1 by 4. Then you will go for 1 by 8 as the answer. But they have given that homozygous is lethal. Only there is a possibility of what? Your FF and FFG. Okay, out of 3, 1 by 3 will be your what? FF, and this will be your 2 by 3. So 9, 2 by 9. Sorry. There is some confusion here. What is the total frequency of this particular FFG and FF? Okay. One by 2 into there is 1 by 3. Yes. This will be your 2 by 3. This will be your 1 by 3. There will be a 3 progenies. Out of those, the frequency of heterozygous will be 2 by 3. And this frequency of this normal feather will be 1 by 3. Since there are no the lethality, there are no FG progenies. Okay, then you should multiply with 1 by 3. So what will be the answer? 1 by 6. This is the frequency. Okay, you should be very careful. They have given that this particular genotype will be lethal. So you should not consider that in the frequency. So that out of these two, 2 by 3 will be your particular allele and this one will be 1 by 3. Okay, this is the answer. Once more explain. This is the condition of the question. Anyway, question you already got it. They are asking for pink colored neck, frequency of pink necked and normal feathered. Okay, pink necked condition, that will be your NR, NWS, since they are having independent, two are independent genes. Okay, that's why it is two different traits. Your NR, NWO will be 1/2 frequency since it is heterozygous. Okay, if you see 1/2, this is your pink colored neck. And what about normal feather they are asking? Okay, in this case they are telling it will possess, FF will possess normal, FFG produce globe feather, and homozygous FG is lethal. Okay, whatever the genetic condition where FG, the your bird will going to die. There is no frequency in the population. But in the remaining FFFG and FFFF, okay, if you see out of this particular thing, the two progeny will get like this, and one will get like FFFF. The remaining gametes will produce. The remaining constitution will be two will be FFG, one will be FF. So out of, if you want to take the frequency, 2 out of 3 will be FFG, heterozygous condition, 1 out of 3 will be FF. You need this particular one, that is your normal feathered birds. They are asking for normal feathered birds. Okay, so 1:2, 1/2 into 1/3. So I will get the frequency 1/6. Okay, yes, haplolethality. We can call like that. Which of the following NGS technology works on polymerase based real-time sequencing? You should know the principle. Maybe they can ask. Okay, definitely they will ask based on the principles only if they are asking any NGS technology. Okay. B, solid. B, sir. No answer. Sorry. I am seeing this particular one. D only. Okay, your 4 sequence is based on pyro sequencing, based on light detection. Okay, I have explained each and everything in our live classes. Solid, it is based on ligation based technology. Okay, ligation based detection, we can tell. Illumina, it is reversible terminator technology. Okay, these are the different cases. But your SMRT is polymer based real-time sequencing. Okay, it contains the micro zapaloids. We will call there's a zero mode waveguides. They will detect. Okay, those waveguides will detect immediate and real-time base pairs. Okay, your answer will be SMRT. A wheat genotype was found to be resistant to powdery mildew in Himachal Pradesh. The F2 population was developed with contrasting susceptible parents. Okay, what they have did, they got one genotype which is resistant to powdery mildew. Then they developed some population with the contrasting susceptible parents. The F1 plant observable to be resistant. What are the data they are giving? F1 is resistant. Then in F2 population, 384 and 296, out of 384, 296 were resistant and 88 were susceptible. So you should calculate F2 ratio, at what ratio it is segregating. That is very important. Okay, the F2 ratio. Additionally, in 96 backcrossed progenies with susceptible parents, 50 were resistant and 46 were susceptible. Additionally, in 96 backcross progenies, backcross with the susceptible plants. That means backcrossing with susceptible, that is nothing but your test cross. Is it? The test cross. They are giving test cross ratio with susceptible parents. 50 were resistant, 46 were susceptible. That means 1:1 ratio. They are giving test cross ratio. They are giving too much information. Based on your F1 and F2 ratio only, you can decide, okay, the genes, okay, whether they are in which condition. Okay, Bhavesh B, sir. which you are for this question? Yeah, yeah. Okay, we will see. Further, in 96 BC1, developed through selfing, 96 BC1 F1 plants, about 23 families from susceptible BC1 F1 were segregating at a ratio of 1 or 3. Yes. Okay. Out of those susceptible BC1 F1, okay, if they go for again for the next generation, how they are segregating? 1:3. 1 resistant, 3 susceptible. This is what they have given. Okay. Here, anyway, they have already given that 1 dominant, 1 recessive, 2 dominant, 2 recessive, 2 dominant. They are talking about 2 genes. Okay, there is no chance for 1 gene. They have already in the options only they have given that there are possibility of 2 genes. But how what is the combination of those genes? 1 is dominant, 1 is recessive, 2 dominant, 2 recessive, and complete linkage. अरे भाई इसमें देखो F1 they are giving, it is resistant. If it is resistant, okay, you can easily neglect that the 2 recessive resistance genes. If it is the 2 genes are resistance, then how your F1 will come resistance? If 2 recessive, sorry, if the genes are both the genes, okay, which are having recessive genes will control the resistance, then how you will get F1 as a resistant? You can easily neglect. You can easily remove this one. Okay, then the remaining are 2 dominant with complete linkage, 2 dominant independent, and 1 dominant and 1 recessive. So that you should know the different ratios. Okay, how it will segregate. In case of digenic, when there are 2 genes responsible, if both are dominant, F1 will be resistant. Okay, and in the segregation, 15:1. Okay, if 2 dominant genes, okay, it will segregate as your normal dihybrid cross, 15:1. Okay, when both are dominant. But when there is a one dominant and recessive condition, you can see your F1 will be resistant since any one of the copy is just needed to give the resistance. So your F1 will be dominant if it is one dominant and one recessive. But the segregation in F2, it will be 13 are resistant and 3s. It will be segregate like this. But in case of both the recessive, as I told, you can decide in the F1 itself. If it is susceptible in F1, then it will be recessive only, both the genes. Okay, there is no need to move for further. Okay, if you move further also, in F2, you will get 7 resistant and 9 susceptible. This is the condition. Okay, now we have to see these two, both dominant. In the options, they have given 1 dominant, 1 recessive, 2 dominant and 2 dominant resistant. When this is the condition, 2 dominant resistance genes with the complete linkage, when there are 2 genes with the complete linkage, then there is no segregation. They will behave as a single gene. So that what you will get in F2, your ratio will be 3:1, like single monohybrid ratio. Okay, if it is complete linkage. So your F2, they have given 296 and 88. 296 and 88. It is not 3:1. We can easily say. Okay, it is not 3:1. If you take, it is around. If it is 3:1, then it will be around like 270 and 90. But it is 296 and 88. Okay, it is not at all related to or near to 3:1. Okay, we can neglect. So there is no complete linkage also. Then there is if the both are dominant, then it should be 15:1. अरे ये तो 15:1 जैसे दिख रहा है क्या? नहीं. Okay, then if you calculate, it will come around 13:3. The very near to 13:3. So that you can tell that it is controlled by 1 dominant and 1 recessive gene. If you move further, so that you can test it in the test class also. And they are given that, okay, they have moved further and they are in BC1F1. From the BC1F1, they are taking 96 BC1F2 families. Out of that, again, they are seeing that selfing of 96 BC1F1 plants, about 23 families from susceptible, there is a segregation ratio of 1R into 3S. Okay, if your BC1F2 is again segregating in the 1R in 3 susceptible ratio, that means, okay, you are having one dominant and one recessive. Otherwise, it will be segregated as 3:1. Okay, if it is two dominant genes, as I told, but it is in 1R 3S. So you can easily tell that it is based on the one dominant and one recessive. Okay, don't think too much about it. Okay, you can see based on, forget about your test class and back class, everything. Based on your segregation F2 ratio, you can decide. Okay, if it is 3:1, then single gene or two genes in complete linkage. Okay, if it is both dominant genes, when there are two genes, 15:1 ratio, one dominant, one recessive, 13:3 ratio. Both are recessive, then 7:9 ratio. And the susceptibility in case of F1, if it is both are recessive. Okay, clear? So your answer will be one dominant and one recessive gene. Then this is about our courses. Okay, so many students already purchased our courses and they are preparing for this particular current examination. Okay, now I will not recommend you guys to purchase any courses for this particular exam. Anyway, there are only 7 or 8 days left. This is for next year. We have launched our PhD course for 5,999. Okay, test series will be 8,999, and for ASR Minute and ICAR PhD, both will combine. Okay, you will get it at 6,999. Okay, this particular course will be also included in this particular ASR Net course. Okay, this will be valid till your examination, the next year 2027 examination. This will be valid for 365 days, and the test series you will get it at 799. Okay, I am not telling you guys that you can directly purchase our courses. You can take feedback from our previous year students. Okay, based on that you can purchase our courses for your preparation. Okay, then the central limit theorem states that there are different options. The sum of large number of independent random quantities always diverges from the normal distribution. It is regarding the samples, random samples. Okay, if you go to the field and you will start to taking the random samples, okay, then you will measure the values. What you will do, you will go to the field and you will choose some 5 plants and you will start to record the data of plant rate, for example. If you keep on doing those random quantities observation, okay, whatever it may be, the population, I will not care about your original population. You will start to do some random measurements, random sample measurements, and you will keep on doing like that. After that, if you sum of the large number of independent random quantities, you will get a normal distribution like this. Okay, I am not asking you that in normal distribution population only you need to go and do the sampling, whatever the original population or whatever the original data set. If you start to do the independent random quantities, okay, if you make the large number of those particular random quantities, then if you do the summation, then you will get a normal distribution curve. Okay, for example, if you go and do the any data analysis or any collection of data of the random, if you choose some random people and you will try to take the their height. If you keep on doing that, okay, then at last if you see the data, then it will be like normal distribution. Okay, the sum of the peoples, like the group of people will contains less height and it will go like this, the majority of people will having the medium height, then again there will be if you keep on increasing the height, the frequency of the individuals which are having that particular height will be decreased. This is the condition. Okay, whatever it may be, the conditions will be like that. That what exactly our central limit theorem will tell. The sum of large number of independent random quantities always converges to a normal distribution. C, good. It is C. Then in triple test class analysis, the L1 plus L2 minus 2L3 values given resulted in estimate of minus 12.56. Which of the statement is true with respect to TTC analysis? Statement 1. The means of L1, L2, L3 are the mean values of progenies derived from the crossing between parents L1 and L2 and L3. That is your F1. Statement 2. TTC value, this particular value indicates the absence of epistatis. बताओ, आंसर बताओ. Yes? What is the answer for this particular question? C. Good. Statement 1 is true. Okay, your L1, L2, L3. This is the mean value of P1. This is the mean value of P2 parent. This is the mean value of your F1 parent. Your TTC is extension of your NCD 3. Okay, North Carolina Design 3. Extension will be your TTC. Okay, if you do that L1 plus L2 minus 2L3, it is 0. If it is 0, then there is no epistatis. Okay, but any deviation from the 0 shows that there is a presence of epistatis. Okay, so your statement 1 is true, statement 2 is false. This is your answer. Okay, it indicates the presence, okay, of epistatis. The sweet genes are part of family of sugar transporters in plants. And statement 2, bacteria causing bacterial blight can utilize sweet genes to assess nutrients leading to susceptibility. The statement 2. What is the answer for this particular question? Statement 1 is stating that sweet genes are part of family of sugar transporters. Those sweet genes are utilized by bacteria to leading that will leads to the susceptibility. Yeah. Yes, exactly right. What is exactly the sweets? That means are the sugar will eventually be exported. Transporters. Sugar will eventually exported transporters. The full form of sweet. These are the genes which are responsible for sugar transporters in the plants. Okay, those will be utilized by the bacteria. Okay, bacteria is having some transcription activator genes. What it will do when it infects, okay, those transcription activator like genes will enhance this sweet genes production. So there will be there is a production of this particular sugar transporters. So that plant, the bacteria can access to the nutrients. Okay, the sweet genes, so that it can also access to the nutrients. The more production of nutrients, the sugars, it will leads to the susceptibility. Okay, both statement 1 and 2 are correct. Telomere functions to protect the end of the chromosome. They are asking for the correct statements. Okay, yes, it will protect the end of the chromosome by telomerase enzyme. Telomere function is allowed to telomere to be extended. Yes. Okay, it will be extended so that in the next replication, if it is in the pre-termination replication, it can be maintained. Telomere facilitates meiotic chromosome reorganization for efficient pairing of homologous chromosome. Yes, it will help in the efficient pairing mechanism. The telomere facilitates intron splicing. Okay, there is no involvement of your telomere in intron splicing. Okay, the answer will be A, B, C only. The next question. Parental lines developed via double applied method were crossed to maximize heterosis in maize single cross hybrids. Yes, we will use double applieds to maximize the heterosis in case of single cross hybrid. Consideration of heterosis, that is your HF1 is sigma dy square. The difference in gene frequency y between them, between the double aploids, what will be the y value with respect to this particular formula? Y is nothing but the difference in gene frequency. What can be the value? What can be the value in case of this particular question? Y. Anana Patra, C. For which question you are telling? For this one or previous one? Yes, the answer will be most repeated question, sir. Yes, during our time also they have asked why, I don't know, maybe why in 2022 paper. Yes, what will be the answer? Yes, jaldi jaldi. It is you precious time for you guys. 0 or 1. Okay, when it will be 0 or when it is 1. Okay. When you are inbred due to applied, okay, capital A, capital B, C, D, E. B, C, D, E. And then another inbred will be, sorry, capital A, B, B. There is a complete difference, okay. Small a, B, C, D, E. B, C, D, E. This is your homozygous. Maybe through double applieds you can get homozygous like that. Okay, in this particular condition, your Y will be 1. You can expect your difference, that gene difference will be 1. In another case, your both double aploids having the same condition like this or like this. At that condition, your gene difference will be 0. If it is both are the same gene, there is no difference at all, then there will be 0. And the other conditions also like this you can expect. Okay, the sum of the genes are capital A, small b, capital C, small d, capital E. Okay, like this. And in case of your another inbred, small a, capital B, small c, capital D, small e. You can see the difference. The difference will be 1, complete difference. If it is capital A here, small a here. If it is small bb here capital BB. Here also you can expect y is equal to 1. But if both are same, if both inbreds are like this only or this only, then your y will be 0. There is a possibility in case of your double applieds. There are different possibilities. So it will be 0 either or 1. Okay, clear? Batao? Clear? Is it clear? Yes or no? इसलिए मैं यूट्यूब में क्लास लेने के लिए उतना इंटरेस्ट नहीं होता है मुझे, इंटरेक्शन नहीं होता है अच्छे से. Then match the following: 1 factor design, 2 factor design, 3 factor, 4 factor with the different mating designs. Okay, in case of top cross, this will be your inbreds. There will be so many inbreds. You will cross with the common any OPV or anything, your tester with the tester. The tester will be common. You are only testing this first set of inbreds. So this will be your 1 factor design, top cross. Then in case of line into tester, what you will do? The lines are one set, your testers are one set. Okay, L1, L2, L3 into T1, T2, T3. There will be different combination. So that will be your 2 factor. 3 factor will be your triallele. You will use 3 inbreds in the cross. 4 factor your quadriallele. And the components from this particular process you are getting. Based on that also you can differentiate 1 factor, 2 factor, 3 factor, 4 factor. In case of top cross, okay, there is only 1 factor, genotype. In case of line to tester, you can go, there will be 2 factor. Okay, variance, additive variance, dominance variance. Okay, in case of your triallele, you can go for 3 factor, dominance, variance, additive variance, and epistatic variance. Okay, then in case of 4th factor, that is in quadriallele, okay, again there will be a 4th factor, how they are interacting. You can see complete details. Based on that also you can differentiate these two. 1 factor will be 4. There is only one option. Okay, you can easily choose this particular option. Okay, B for 3. Yes. Choose the correct answer from the options given below. There will be a, these are like LD estimates. Okay, what you know, R square. This we will commonly use in case of our GWAS. Okay, so that you can easily tell that most oftenly used in the plants. Okay, this is lambda, not A. So for A there will be 2. So you can see A for 2, there are 2 options. Okay, then this lambda, this also you know, this will be used in population genetics. Okay, the lambda, the concept will be same, but we are using the different estimates. Okay, by different methods we are estimating that particular linkage disequilibrium. This will be lambda will be used in population genetics. Okay, D for D1, D1 you can easily choose this one. B3, B3 frequency is in epidemiology. Okay, in epidemiology I too don't know this particular symbols and LD estimates, but based on the what you know, this one R square you know and the lambda you know, based on that you can choose this particular answer. Okay, and your Q will be used in where? Used in case control studies. Okay, genuinely I can say that I know R square and lambda. Based on that only I can choose this particular option very easily. As I told, there will be some questions like this, okay, which are from out of your boundary. Okay, you should be ready for this. Okay, this is a very simple question they've asked. The regarding different crops and introduced from your tobacco from South America, you know, for A3, you can easily choose this one and you can move forward. There is no need to think too much about it. Cherry, B2, yes, cherries. Sorry, cherry from not from China. Cherry from Afghanistan, yes, B from 2. Loquat from China, T and loquat. Anato from West Indies. Okay, this will be your answer. Next question. What is the smallest perfect F2 population size for the 8 segregating and 7 non-segregating gene pairs? Okay, what is the perfect F2 population? That will be 4n. Okay, that n should be, you know, n is segregating loci. Okay, when there are some genes which are non-segregating, there is no need to calculate for those things. They will act as a single gene. Okay, you should calculate for the segregating genes. 4:8. What about 4:8? How much it will come? Anyone? Yes. 4:8. Yeah. What will be the answer out of this? Two 56 into 256 कितना आएगा? Okay. This will be your answer. 4:8. 8. The phenomenon explaining relationship between number of detection of QTL and their effects with the population size. ये भी मैं एक्सप्लेन किया था लाइव क्लास में, ठीक है. Relationship between number of detected QTL and their effect with the population size. If your population size is very less, okay, the detected QTL, the number will be very less. The number of QTLs will be very less. So the each contributing effect will be I. Okay, when you increase the population, you will get a more number of QTLs detected, and each will be accurate values. The effect will be, it will give cumulative effect. But when you are having small population size, okay, small population size, what will happen? You will get detected less QTLs with more effect. You will miss the so many number of QTLs. Okay, it will give the false estimation. This we will call it as a Bayes effect. Yes, good Bhavesh. Yes, Ananna. Organelle or genome are mostly circular molecules of DNA. Yes, your mitochondrial and cytoplasmic genes, okay, which are your mitochondrial and chloroplast which are circular. Organelle genome are transcribed and translated within the organelle. Yes, but in case of your nuclear genes, okay, they will be translated in the cytoplasm, okay, with the help of ribosomes. But here in organelle genome, they will be transcribe and translate both in case of organelle. Okay, both statements are correct. The variance of root number in F1, they have given that 1.97. F2 will be 7.97. They are asking for degree of genetic determination. Genetic determination is nothing but your heritability. They are indirectly asking heritability in the F2 generation. Okay, genetic determination is nothing but your R square value. That is nothing but your heritability. Okay, from F1, if any variation you are getting in F1, that is purely due to environment. That is your 1.97. In F2, it contains both your genotype plus environment. If you minus your 7.97 minus 1.97, you will get how much? 6. That is your genotype. Okay, your heritability will be 6 out of 7.97. This is your heritability. How much? 6 by 7.97. D. Yes. VP minus VE divided by VP. Good. Which one? Answer will be 0.75. Okay. I don't know. I have not seen the answer key. Okay. I will see first. Everything what you guys are telling, I am believing the calculation. 6 divided by 7.97. Yes, that is your 0.75. Heteromorphic self-incompatibility of primula comes under. Okay, there will be two types of self-incompatibility. One is homomorphic, another is heteromorphic. In homomorphic, we will classify into gametophytic and sporophytic. Heteromorphic is based on the flower structure, but the self-incompatibility of primula, which is heteromorphic, which will comes under one of the gametophytic or sporophytic, which is related with the genes are genes controlling the mechanism. Everything will be related. Yes, good. It is related to sporophytic self-incompatibility, but the system is between the floral morphology. That's why we'll call it as heteromorphic. Okay, but it is indirectly sporophytic only. Then choose the correct answer from the option below. Factor analysis, discriminant analysis, canonical correlation, and multivariance covariance. And these are the different categories. Okay, how many sets of variables we are using and population, that sets of different population. Your factor analysis. Factor analysis is nothing but, okay, in the same population with the set of variables, you are trying to reduce the complete complex data set. Okay, you are seeing for the correlated traits so that we can reduce the number of factors into the uncorrelated factors, like your principal component. Here you can whatever the thing, like one set of variables and one set of population in case of your factor analysis. In case of discriminant analysis function, okay, what you will do, you will use the distance matrix. Based on that, you can separate the population. Okay, you will use some traits. Okay, you will use some different sets of variables so that you can make the population into two different population. Okay, where you can use one set of variables, two or more set of population. Okay, variables are same. Like for example, you are measuring for root or different leaf characters. By using that, you are making the population into two: drought susceptible, drought tolerance, based on the single set of variables, the two population. Okay, like this one set of variable and two or more set of population. Your canonical correlation, you are using one or two more set of variables. Okay, the measures, the variables, the traits before flowering and the traits after flowering. You will see the correlation between these two. Okay, the traits before flowering and the traits after flowering, how they are correlating. That is your two or more set of variables in the single population. That is your canonical correlation. Multivariate covariance. Here you will use two or more set of variables and two or more set of population. That is your multivariate analysis. Markova. We will call it as a Markova. So, two or more set of variables with the two or more set of population. Your answer will be yes. The answer will be B. Okay. If you want to see the difference between ANOVA, MANOVA and ANCOVA. In ANOVA, analysis of variance. What is the dependent variable? 1. You are seeing for yield, for example, the variable is yield. You will see the analysis of variance for the yield perspective. Okay. What is the variance due to genotypes for that particular yield and the error. We will make the analysis of variance like this. Okay, there are no extra covariate here. Compare the group means. You are comparing the different group means, or you are partitioning the variance. That is your ANOVA, normal ANOVA. In case of MANOVA, it is multivariate analysis of variance. At particular time, the dependent variables are more than two or more. Okay, in this case, only one your dependent variable. Here, two or more. That is your MANOVA. Compare groups for multiple traits together. Okay, the multiple traits together. In case of ANCOVA, it is analysis of covariance. Usually one dependent variable, but the thing is extra covariate. We will take some other factors as an extra covariate. For example, soil fertility or environment or temperature or rainfall. Okay, it is like an extra covariate we will use in the analysis. Okay, so that we can reduce the error while comparing the different traits. Okay, the multiple traits. Compare groups after adjustment. First, you will adjust the data based on the covariate. Then you will go for analysis. That is your analysis of covariance. ANOVA, MANOVA, and analysis of covariance. Okay, this is the difference between these three terms. Popular baby corn maize hybrid HM4 was developed by. Yes. Baby corn maize hybrid HM4 was developed by which scientist? Sain Prasad, Sain Das, VP Mani, BK Mukherjee. Good. Sain Das. There is nothing related to Sain Prasad. and VP Mani is the responsible for your Vivek QPM 9. VP case or the institute ICR VP case Almora. VP Krushi Anusandhan, yes, VPKS Almora. Okay, the responsible for Vivek QPM 9. And BK Mukherjee, okay, he is scientist from IRI. Okay, he is also expert in maize breeding. Okay, but your baby corn from the Sain Prasad HM4. If money was at the Elm in 2024, BK Prasad will be at the Elm in 2026. Okay. The sequencing of the Chinese spring variety of wheat by International Wheat Genome Sequencing Consortium was performed using ditelosomic stocks. Okay, what are ditelosomic stocks? Okay, it contains the ditelosomic. That means your telosomic chromosomes are nothing but only one arm of the chromosomes are present. Okay, like this. Short arm, for example. Okay, this is like 3B short arm. So there will be a 3B long arm. There will be two different stocks. These are ditelosomic stocks. Okay, they have used these particular stocks for your DNA sequencing. What about telotrisomic, ditelotrisomic, and teloisotrisomic? These are trisomic lines, the extra chromosome, okay, which you are having in the condition. It will be, in case of telotrisomic, the extra chromosome will be telosomic chromosome. There are two normal chromosome. One is telocentric chromosome. In case of telotrisomic, in ditelotrisomic, one will be normal chromosome, one will be telocentric for one of the chromosome, and another one be another arm. This is the condition of ditelotrisomic. What about teloisotrisomic stocks? Teloisotrisomic stocks are, there will be one normal chromosome and another will be, both are isochromosomes. Isochromosomes are teloiso. They are telocentric but iso. Both the chromosomes will be the same arm. Okay, this is your teloisotrisomic. This is your telotrisomic. This is your ditelotrisomic. This is your ditelosomic. Ditelosomic. This is the condition they are used in the wheat. Okay, clear? The pre-sequence sorting signature of protein. Again, the out of the boundary question. Okay, the pre-sequence sorting signature of protein destined or target the proteins into. Okay, this is the pre-sequence. This is one of the protein which will use in the protein translocation. Okay, the proteins whichever they are produced, they will try to move into the mitochondria. Okay, to the mitochondria. A pre-sequence is an N-terminal targeting signal. It is a targeting signal that directs newly synthesized protein from the cytosol into the mitochondria. Okay, what about the other targeting signals? Transist peptide. It will target to move the proteins into chloroplast. Okay, proteins usually enter through the secretory pathway when they want to move into Golgi bodies. Okay, the nuclear localization signals helps to move your protein into nucleus. Okay, these are the different targeting signals. A type of apomixis where the nucleus of the egg cell and generative nucleus divides independently to give rise to the haploid chimera, which is further used to develop double haploid line in the cotton. Batao? where the nucleus of egg cell and generative nucleus will not fuse. They will independently, they will develop, so that you will get an applied chimera. Okay, that is your semigamy in cotton. Okay. There will be a need of pollen egg cell to develop a seed, but they will not fuse. They will independently develop, so that there will be a chimera development. Okay, that is your semigamy condition. Pseudogenes, there will be different statements. They are asking for correct. Pseudogenes are copies of functional genes that have altered or missing region. Yes, it is right statement. That's what pseudogenes explains. Non-processed pseudogenes result from the incomplete duplication or second copy mutation of functional genes. Yes, this is also correct. Pseudogenes can encode endogenous siRNAs. Again, it is true. Your siRNAs, which are endogenous in nature, they will encode by pseudogenes only. Ribosomal proteins, pseudogenes, are unprocessed pseudogenes. This is not correct statement. Ribosomal proteins are processed, okay, are processed pseudogenes, not unprocessed. So your answer will be A, B, C only. The article Molecular Structure of Nucleic Acid, Structural for Deoxyribose nucleic acid, which is published by Watson and Crick, in which journal? In which journal? Batao? Where they have published it? Yes. Batao, koi batao? Yes, Bhavesh. It is in Nature in the year 1953. The particular issue, I forgot that issue and volume name, I think, in the 1953 in the Nature journal. It is for the paper by Watson and Crick and paper by Wilkins et al. And Roslin et al. All are published in the same journal, in the same issue and the same volume in the 1953. Okay, in the Nature journal. From that era, everything changed regarding the DNA. Which of the following multivariate analysis is more suitable when there are a lot of missing data? And the number of traits more than the number of entities in the study. Okay, principal component, principal coordinate. There will be always confusion between principal component analysis and principal coordinate analysis. You should know that principal component analysis is based on correlation matrix. Correlation matrix means it will see for each values in the traits and each values in the another traits. It will try to calculate the correlation between these two values. Okay, so that it is like correlation matrix. If there are missing values, then it will be difficult in case of principal component analysis. But in case of principal coordinate analysis, it will be based on similar and dissimilar matrix. It will calculate the distance between the two entities or two values, so that if there are any missing data also, it can manage. Okay, your principal component analysis uses raw trait data, but it will use distance or dissimilarity matrix. Based on that, there will be a principal coordinate analysis. Based on variance and covariance or correlation matrix, it is distance among entities. Needs cleaner complete data. It can handle missing data also. Shows major variation in the traits. Okay, it shows dissimilarity or similarity among the entities. Principal component analysis will use principal coordinate analysis. We will use to see the based on dissimilarity similarity. It will divide. It will divide the population. Okay, where there are missing data. Okay, and also the number of traits is more than the number of entities. Okay, for example, entities are like observation, how much you are taking, the values. Okay, but the traits are more in this case. The traits are more and your samples are very less. In that case, okay, you will go for principal coordinate analysis. As I told, it will based on the similarity or dissimilarity matrix. Okay, there is no, if there are any missing data also, it can manage. Which of the following methods offer early segregation or early generation testing of families? early generation testing of families. Yes, among these options, which of the following methods offers early generation testing of families? C. Bhavesh. Okay, what about others? Grid and pedigree. Okay. Then there are only two peoples. There are there are only two people they are watching this live. There are no other. They don't want to respond. As I told it in the beginning only, if the number and if the response is like that, then there is no need to take classes further. Okay, you can see of my classes, PYQ classes and marathon classes in the PG. Okay, how much the response and how they are actively involving in the discussion series, so that I will also get some motivation and it will be like interactive. Otherwise, I can teach only for Ananna and Bhavesh. Grid and pedigree. Okay, all. Okay, all of the above. Individual and mass selection. What we can do in case of mass selection also, based on phenotype, we will go for early based on different families. You can reject it in the early segregation. Your grid, Robic grid, honeycomb, these are involved in the pedigree selection only. Okay, after F3 or F4 progenies, okay, based on that, you will try to evaluate the families within the family selection. It is based on the grid, honeycomb, whatever it may be. It will comes under the pedigree selection only. Okay, what about grid selection? This is your field. Okay, you will try to make a different grid. In mass selection also it will come your grid selection. You will make a will divide your plot into different based on soil heterogeneity. Will divide into field into so many parts, and you will try to select the plants within that particular grid. That is your grid selection. What about Robic grid? Your plot will be, in case of Robic grid, plot will be in the diamond shape, like this. कंफ्यूजन हो रहा है diamond लिखने के लिए, like this in the diamond shape. In case of honeycomb, it will be hexagonal shape. Okay, based on that, we will try to select within those particular shapes and we will compare. After that, we will try to compare within that particular grid, not between the grids. Okay, based on that, we will select, and in the final step, based early in the early generation, we can remove early in the we can do the selection in the early generation like this. Then in the final selection, we will compare between the grids or between the honeycombs. Okay, this is what early generation testing. Okay, we will do all these in particular condition. Okay, Bhavesh, clear? You are only telling grid selection or honeycomb selection. These are the parts of your pedigree selection. Okay, you can use it in pedigree or mass selection. Okay, clear? Any confusion? Okay. The next, EFR, again out of the boundary question. Don't think that. Okay, there are so many which are related to your stress breeding. That too disease resistance. Okay, maybe the person who went for this paper setting, maybe he is from the specialization in the breeding for disease stress. Okay, so many questions you can so that maybe you are expecting so many questions from this particular part. EFTU receptor is leucine rich repeat. Okay, receptor like kinase. Statement 2, EFR is associated with the effector trigger immunity. Okay, if you don't know this particular concept, then definitely you are not able to attend. मतलब कैसे आपको पता चलेगा ये तो इम्यूनिटी ये सारे रिसेप्टर्स ऐसे, ठीक है? Like these questions, no one will correctly answer. Maybe they are guess, maybe they have choose the options through guess, so that maybe they will get an marks. But these are somewhat like out of the boundary questions. Okay, what about, yes, this is the statement is right. The EF receptor is leucine rich repeat, and EFR is associated with effector trigger immunity. This is not a correct option. Statement 1 is correct. Statement 2 is incorrect. Okay, this is the your right answer. What exactly the effector trigger immunity? Okay, you can see here. This is pattern triggered immunity. It is effector triggered immunity. In pattern triggered immunity, what will happen? Triggered by pathogen associated molecular pattern or microbe associated molecular pattern. Okay, in the cell membrane itself, there will be a receptors like EFR. They will recognize this particular pattern when your pathogens will attack. Okay, in the cell membrane itself, there will be a first line of defense. Okay, broad spectrum resistance. But in case of effector triggered immunity, it is triggered by pathogen effectors like flagella. In case of bacteria, there will be some effectors, okay, which are related to pathogen. Receptors are inside the cell, here in the cell membrane. These are second and stronger defense. Once the molecule or the insect, sorry, the pathogen try to establish, then there will be a specific response. Okay, it is res specific. It is general mechanism. Your pattern triggered immunity, effector trigger immunity is specific and stronger defense. Okay, this is again regarding too much about into pathology. Okay, maybe you can expect these kinds of questions, and this will be not repeat always. Don't think too much about it. Genetic situation where AA, BB, AA, capital B, small b, AA, B, B produce single phenotype class, and where both dominant alleles, capital A, capital B, are present together, produce another phenotype. Okay, it is 9, 3, 3, 1. That means 9:7, where you can expect this particular interaction. Yes, guesswork, Bhavesh. Yes, some questions through guesswork they will get marks. Okay, we can't do anything. Sir, complementary gene action. Yes, that is your duplicate recessive interaction, where there is any one condition is recessive. Okay, you can see here there is a recessive condition for this particular allele. There is a recessive condition of this particular allele. Here both are recessive. At that condition, you will get another phenotype. Okay, in the remaining the one class you will get this. So that's why we will call it as a duplicate recessive. The minimum any one of the gene in recessive condition, there will be a different phenotype. In case of duplicate dominant, what will happen? Okay, any one gene, if it is in the dominant condition, then there will be a different phenotype. Okay, at that particular condition, okay, you can see this one, any one is in dominant condition, this one and this one, 2 are. So 9, 3, 12, 3, 15. 15:1. This is your duplicate dominant epistatis. In recessive, any one is recessive, then there is a different. In duplicate dominant, if any one gene is in dominant, then it is duplicate dominant. Yes, good. The answer will be in this condition, duplicate recessive. The ultra low GI rise developed using IR36 AE lines as a GI value of. This is based on the irrephilipines. They have made the different categories. Okay, again this you can't expect. Okay, how you can expect this question will come? The ultra low GI rise. Okay, yeah, answer will be 45 or less. There will be different classification. Okay, before less than 45, 46 to 55, 56 to 80, and 80 to 100. The different classification: ultra low, low, medium, high, like this. So, okay, the less glycemic will be the low, ultra low will be less than 45. Okay, this is the classification. What about what is glycemic index? It is nothing but based on amylose and amylopectin content. Okay, if it is more amylose, less amylopectin, okay, that is glycemic rise. There will be less glycemic index. Okay, if it is more too much amylopectin, there will be too much sugar, so the glycemic index also will increase. Okay, the ultra low glycemic index, why we need it? It is for sugar or diabetics patient. Okay, you should maintain ultra low GI index so that there will be a less sugar in the rice grain, so it can avoid the diabetics. Okay, the virus free stocks in meristem culture could be obtained through which of the following treatments or approaches? Cryotherapy, yes, by using -196 degree temperature. Chemotherapy, by using antivirin chemicals. Okay, it will kill the viruses. Then thermotherapy based on the temperature. But phage therapy, अरे भाई, phage therapy मतलब क्या है? phage, वो virus, they will attack bacteria. It is not regarding the virus free stock meristem. Okay, yes, ABD only. NES pistillate lines of a castor maintains through. It is completely reverse for your TGMS. In case of TGMS, what will happen? It is around more than 24 or okay, more in higher temperature. Okay, there will be sterility, lower fertility. In case of your TGMS, there is a particular cutoff temperature, right? How much it is in TGMS? The cutoff temperature, Bhavesh? In TGMS, I am asking, normal thermosensitive genetic malleability. What is the cutoff temperature? In case of rice, yes, rice for example. बताओ, कोई बताओ. 26. Maybe you are also telling maybe. It is around 24 point something. Okay, more than that particular temperature, you will get sterility. Less than that particular temperature, it is fertility. But in this case, growing in the environment at above 31 degrees Celsius, more than that, okay, it will act as a normal fertile plants. That means you will get both staminate and pistillate flowers. Okay, so that you can maintain. If it is less than below 30 degree, then you will not get staminate flowers. Only you will get pistillate, so that your plant will be completely female. Okay, your answer will be A here. If you grow in the above 31 degrees Celsius, so that you can get both pistillate and staminate flowers in the same plant, so that you can maintain. If random chromosome assortment occurs in the autotetraploid in the duplex condition, capital A, small a, small a, the gametic ratio, they are asking for gametic ratio from this particular plant. Okay, if you take the normal condition, they're not telling any exceptions here. Normal condition. So from out of this particular tetraploid, you will get diploid gametes. Those are like capital A. Okay, there is only one chance of capital A combination. So in one condition, here this capital A may be fused with this one, this capital A may be fused with this one, or this with this one, this with this one. There are 4 possibilities of capital A, small a. Okay, then the small a will be only single condition, small a, 1. Okay, what I am explaining here, how we will get 4 capital A, small a. You can see this capital A with this with this, okay, and this with this with this. There are 4 different combination. Okay, that's why we will get this. The answer will be first one. The optimal selection intensity for selecting trait A, trait B is 0.05 and 0.01. This is the optimum for any to select any traits. The intensity, the selection intensity. The suppose selection intensity matlab if you are it is 0.005, that is 5% of the population you can select for the trait A. 0.1 means the 10% of the population you can select. Okay, this is the optimal selection intensity for different two traits. They are asking if geneticist wishes to select both the traits simultaneously from the same population, what could be the probable better selection intensity? If you are to multiply 0.05, if you want to select both the trait, so you will get around 0.005. What it means? 0.005, that is 0.5% of the population. It is very difficult to select 0.5% of the population. Okay, out of, for example, out of 100 it is 0.5. Out of 1000 you have to select only 5 plants. If you select both the traits, okay, it is very much difficult. So you should relax your selection intensity. Okay, you should relax. Out of all these 4 options, if you want to relax the selection intensity, that you can do it only through this particular first option. Okay, others are again the intensity will be very less compared to the basic original intensity. In original intensity, you can see only 0.5% population you have to select. It is very much less. Okay, if you want to relax, then you should go with this first option. You can see all other options. By seeing only, you can tell that okay, it is less selection intensity compared to original. Okay, इसलिए the first option will be right answer. Simple. You should be think like this. Which of the following statements are correct with respect to the functional markers? Functional markers are nothing but the markers which are located within the genes. Functional markers derived from the polymorphic sites within the genes affecting the phenotypic variation. Development of functional markers require allele specific sequences of functionally characterized genes. Yes. The use of functional markers in population breeding approaches avoids genetic drift for the characterized loci. Okay, the use of functional markers in a population breeding approaches avoids genetic drift. Yes, if it is functional marker, then it is having advantage over the genetic drift. Okay, you can characterize that particular genes. There is no loss of that particular markers in the future generation. Functional markers derived from the such motifs which can be used to fix gene or alleles in several genetic backgrounds without additional calibration. Okay, all are correct regarding to functional markers. SDN इसलिए आया था लास्ट टाइम, ये रिलीज हुआ था 2 वेराइटीज जीनोमेटेड. Okay, इसलिए पूछा गया है क्वेश्चन. SDN2 involves template guided repair of targeted DNA. Yes, SDN1 will be non-template, only non-homologous end joining. In SDN2 there is a template guided repair. In SDN3 there is a complete introduction of exogenous or foreign DNA. This you should know. Then your SDN1 and 2 are accepted. Okay, SDN1 and 2 are now accepted under the Rule 20 of the 1989 rules. Okay, but here they are telling SDN2 genome edited plants are not accepted. Okay, SDN1 and 2 are accepted. Okay, your answer will be statement 1 is correct and 2 is incorrect. Good. Good, Anana Patra. Which of the following statements are true with respect to honeycomb selection? As I told, honeycomb is nothing but hexagonal. Hexagonal aa jao bhai. Okay, this is your thing. This is your central plant. You will compare with hexagonal, the 6 plants. Okay, and you will based on that value you will try to select. It increases the efficiency of early generation testing. Yes. Nullify the effect of soil heterogeneity on selection. This is the plant you are going to select. You will compare this with the surrounding so that you can nullify the effect of soil heterogeneity. You are not only selecting based on this individual plant, you are comparing with the surrounding thing. Yes. Allow selection only within families. No. It will also allow between families, within and between both. It ensures no competitive effect. Yes, your answer is A, C, D. A, C, D. In digenic interaction between two independent genes AA and BB, the genotype of AABB is crossed with genotype of AABB. Then back crossed. That means your capital AA small. First they are crossing capital AA BB into small AA BB. You are getting capital A small a BB. This is your F1. Then back crossed with the parent with small a BB. If you back cross this with cap small a BB, small a BB, you will get what you will get. There will be different gametes, capital A. So you will get capital A, capital B, small b, small a, capital B, small b. Okay, capital A, small b, small b, small a, small b, small b. These are the test class progenies. They are asking following method designation, that is A, D. Okay, additive and D is dominant. What will be the coefficient of the homozygote into homozygote interaction? This will be in the 1/4, 1/4 into test class, 1/4, 1/4. Okay. Homozygous into homozygous interaction of DA and DB in the backcross generation. Okay. DA means they are asking additive variance in case of A gene and additive variance of B gene. Homozygote, they are asking homozygote into homozygous. In these, out of these 4 test class progenies, both are homozygous. In this particular case, what is the value of small a minus D, small b small d into minus D. Okay, minus D into minus D. You will get plus D value. What is the frequency? 1/4 plus 1/4. Clear? भावेश, अनन्ना, बस आप दोनों है, मैं आप दोनों के साथ बात कर रहा है. Clear? How it will come 1/4? The PP of FRA 2001 gives protection for field crops, which is for 15 years, 18 years for trees and vines. If it is 15 years, then 18 months. In general, for every 1% of error rate in the genotyping, marker adds 2 centimorgan of the inflated distance to the map. If it is 1% error in the genotype marker, then there will be 2 centimorgan inflate. There will be a disturbance in the map. Each error in a non-terminal marker causes 2 apparent recombination in the data set. Yes, your reason is correctly explaining. Both A and A are true, and it is correctly explaining the A. How you can say there is a marker 1, marker 2, marker 3. They are telling that each error in a non-terminal marker, non-terminal, that is M2. If you are talking about terminal markers, then it is different. They are talking about non-terminal marker. Marker is not in terminal region. M2. If there is any error in this particular marker in the genotyping, okay, it will affect the combination with this one also. It will affect the combination of this one. If it is only one recombination, then 1 centimorgan. It is also affecting the error in this particular recombination. So there will be a 2 recombination, 2% of the recombination. So there is a 2 centimorgan inflated distance to the map. Okay, if it is in the non-terminal region. Okay, this is about the explanation. This is all about the 50 questions which are appear in the 2025 Part C paper. I hope you liked this discussion. Okay, I have given clear-cut explanation for each of the questions. Okay, I have not skipped any questions or any doubts regarding to particular session. Okay, if you guys like this, sir, first grand mark test before 4th. Okay, Bhavesh. I will try. I will definitely try. Okay, if you guys like this session, okay, I need that like and comment. Comment तो जरूरी करना है, ठीक है? Based on your comments, I will try to go for another paper discussion. Okay, maybe whether it may be part B or 2024, 3, 22, like that. I will try to cover all of the papers before your exam. Okay, but it is again based on your comments and based on, okay, the views. If you the views are like this and there is no one interested to learn or to discuss these papers, then no worries. Okay, anyway, I have too much views and the love from PG students already. Okay, and anyway, our PhD students also knows about me. Okay, how we have covered each and everything in the live classes. Okay, it's about you guys. Okay, I need comments and share and also need to views. It is just, I will not get anything from your comments and views. Okay, what I will get, the motivation, so that I can take up another classes. Okay, this is about 2025 paper discussion. Okay, all the best for each and everyone for your exams. And one more preparing for next year, I have already told about our courses. Okay, based on the feedback of the students, you can enroll to our courses also. Okay, this is about today's session. Thank you very much. We will meet you in the next class.